Welcome to the Australian Biocommons webinar series. Through these sessions, we hope to share useful information about the latest digital techniques, data and tools. Our goal is to support Australian bioscientists to deliver their best environmental, agricultural and medical research by offering a platform for local and international peers to discuss their work. My name's Christina Hall and I'm the Biocommons Training and Communications Manager. We particularly appreciate those of you joining us live today, and you'll have the opportunity to ask our speaker questions via the Q&A function on your dashboard, and these will be addressed at the end of the presentation. This session will also be recorded if you'd like to revisit it in the future. You'll find it on our YouTube channel, along with recordings of previous webinars and workshops. We also hope you'll keep in touch to hear about future webinars via the channels listed on the screen. Before we start today, we'd like to acknowledge the traditional owners of the lands on which we work and pay our respects to the elders past, present and emerging. Our speaker today is Michael Franklin. Michael is a research software engineer working on the Portable Pipelines project, a collaboration between Peter MacCallum Cancer Centre, Melbourne Bioinformatics and the Walter and Eliza Hall Institute. Michael joined the project in 2018 after graduating with a Bachelor of Science and Bachelor of Computer Science degree from Monash University. His primary research interest is making bioinformatics analysis pipelines more portable and reproducible. And his project leaders are very pleased that Michael will continue on with the project for another year now. Today, Michael will provide an overview of Janis. So I'll invite him now to share his presentation. Welcome, Michael.
Thanks for the introduction. I'm here to talk about Janis and portable pipelines and how you can build once and run everywhere. The slides are available online, and I believe the presentation and the slides will be published later as well. Cool. Today we're going to talk about portable pipelines and workflow systems and how they interact and how you can use them to build your analysis. We'll talk obviously about Janis and one of our solutions to these problems, and the Janis Assistant, a tool that we've built to help with this process. And we're also going to demonstrate Janis. We're going to build an alignment workflow, and we'll run this workflow that we build using CWL tool and Cromwell, which are two execution engines that exist in the workflow space. But we'll talk more about that soon. The process we're going to talk about today, the area is mainly honing in the secondary analysis, a part of the bioinformatics process. So here we've already done our experiment, we've got some data, and we're really kind of doing our alignment, variant detection, annotation, and it's before our tertiary or downstream analysis. So we're just going to focus our area on there. But let's talk about the Portable Pipelines project first. It's a collaboration between Peter Mac, Melbourne Bioinformatics at UniMelb, and the Walter and Eliza Hall Institute. The goal is to produce portable and publishable analysis, a goal with a flexible execution environment. So when we write our analysis, we don't want it to be locked to a single institute. We want to be able to share this and run it everywhere. We want to be adhering to community standards. So we don't want to follow the community best practice, and we don't want to redefine anything brand new. And we want to make it uncomplicated for non-pipeline experts. So it's easy to build and run pipelines with minimal configuration. So some of the considerations when we're building our analysis is how it might scale. So whether we get bigger and bigger data sets with a higher depth for sequencing, or we just get more quantity of analysis. We want to consider how we're going to link multiple pieces of tools together, especially when they're complex bioinformatics software. So we might use workflows or pipelines to do this, and we will use workflows and pipelines to do this. We want to consider how we're going to build and execute our pipelines, whether we're going to be tied to a specific set of infrastructure and really optimize our workflow, or maybe we want it to run on local HPC and cloud environments. And that's going to be important when we share our workflows. One other consideration when we're sharing our workflows is how we might publish these, so what format we use. And we're also going to embed containers to ensure reproducibility across the different execution environments, especially when we're sharing. So we talk about workflow systems, and a lot of researchers already have bash scripts, and they have a variety of different tools that they've bashed together to make work. But with this new set of analysis in this project, we want to consider workflow systems. And these workflow systems are used to connect different tasks together, and it's based on dependency resolution. So what we say here is one of these variant callers, which are these blue bubbles here, they rely on the output of an alignment step. So then there's an edge and a dependency based on that. And in this diagram, which is generated through Radix Composer, a CWL tool, a community tool, the blue steps are the blue nodes are the steps, and the gray steps are inputs and outputs. And this is what a visual representation of what a workflow might look like. So some advantages of using workflow systems over maybe just simple bash scripts, or we get restart, continue, and rerun functionality. So if our workflow fails halfway through, we may be able to tweak some of our inputs and rerun it from where we left off. Workflow systems also give us a greater audit trail and notifications. So we can see who ran the pipeline, how long each step took, and all that's done automatically. And it can notify you when the pipeline succeeds or fails or moves into a different state. The workflow systems that we talk about today can do input and output checking, including the return code. So if your tool doesn't run correctly and the outputs that you expect aren't present, then the workflow can fail immediately and tell you, rather than get a couple steps down and then fail for some unrelated reason. The workflow systems we'll talk about today are abstract from the batch system. So the workflow and the analysis that we build doesn't directly interact with the batch system like Slurm or PBS. They use per tool resources, so we get a better utilization of CPU and memory for our overworked workflow. The systems are abstract from the file system. So we don't talk specifically about paths on a shared file, but we talk about inputs. Our tool will ask for specific inputs and provide outputs. And this helps us generalize to cloud environments just beyond our HPCs. And these abstractions make moving pipelines so much easier around the place, especially to the cloud. So why portable workflows? Well, data is actually really hard to move around, especially when you're talking about massive data sets. It can take a really long time, and it can also be very costly depending on where your data is stored. But also we might want to consider our compute availability. If we get a lot of data sets, it might not be practical to process these all on our local HPC, and we could take advantage of cloud burst computing. Really, we just want our data and our compute to be in the same place. So if our data is stored online, stored through maybe Amazon, we want to move our compute to Amazon. Or if our data is stored locally or at a different institute's HPC, we probably want to process our data there. To summarize this, we just want to bring our pipeline to the data, bring our pipeline and our compute to the data. So the goals of the Portable Pipelines project are to be easy to use. It should use a simple, non-technical API that's friendly to people who aren't pipeline experts. We want to be following the community best standards, following the fair data principles, and we don't want to redefine standards. Our analysis should be portable, and we'll facilitate that using containers and Docker and Singularity container environments. The project should be modular. So by using one component of it, you shouldn't be locked into the rest of it. And it should be extensible. So if you have a better way of doing things, we want you to be able to do that. So some existing workflow systems that we looked at are the common workflow language, which uses a YAML specification, so key value pairs like JSON, to specify your steps, your inputs, your workflows, and your tools. It's fairly tedious to write, but very easy for a computer to parse, as they're just dictionaries. The workflow description language is similar. It's a similar open standard. It uses a custom domain-specific language, which is much easier to write, but harder for a computer to parse. It was initially built by the Broad Institute for the Cromwell execution engine, though there are more engines available now. Some other common existing specifications include Nextflow, SnakeMake, BPipe, all with large community support as well. So some of the advantages of CWL: it's easy to parse. And because it's easy to parse, there are lots of engines that can accept it. And it also means there's lots of community tools like Radix Composer, which was the visualization we showed you earlier, or Benton, which gives you immediate language and IDE support in Visual Studio Code or Sublime. But some of the disadvantages with CWL are it's difficult to learn and write, and there's no meaningful type system beyond file, directory, integer, and so on. WDL has a similar set of advantages and disadvantages. The Cromwell execution engine fits closer to our need. WDL is also easier to write for a person, but it was initially backed by a single institute, which made us a little bit worried. The community isn't as large as CWL, which means there's not as many community tools. And there's also no meaningful type system beyond the file, directory, integer. When we look at CWL and WDL, we get the following execution overview. So we have some specification and potentially some future workflow spec as well. We have a set of execution engines, and you see there's some crossover, and there's also a set of compute environments. Now, not all of these engines work on all of the different compute environments. And even once we've done all our execution, we'll get a different set of outputs, logs, and errors. So we were considering CWL or WDL. Well, why not CWL and WDL? And we could do this by generating CWL and WDL. This would protect us against language deterioration. So for example, if one of the languages lost support on engines, we get a greater flexibility and choice of execution engines, and we can reap the rewards of these multiple community technologies. So we produced Janis. Janis is a Python framework that uses a simple, non-technical Python API to build workflows and wrap tools. It has type-safe connections, and we'll talk more about this in a second. Importantly, it can generate CWL and WDL. And we've also got a toolbox of pre-wrapped tools and data types for Unix and bioinformatics domains. So we talked about the type safety, and it's more than just a file or directory. In Janis, we have special bioinformatics types like FASTQ, paired end FASTQ, BAM, index BAM, and references, and so on, which means when the output of a tool is annotated with a BAM and our second tool is expecting a VCF, we know immediately that this connection probably isn't right, and we can tell you that. Janis supports subtypes. So for example, index BAM, BAM can be received from an index BAM. And talking about index BAMs, there's the concept of secondary or accessory files. For example, index BAM has a .bai attachment, and a lot of the genomics reference files have their different indexes as well. So the toolbox of Janis is a very powerful feature that we've got first-class support for. It's a set of domain-specific tools and data types. And currently we have Unix and bioinformatics tools. These are generally available, so installed automatically, and there's generator documentation immediately available. The example on the right, which is BWMem, a common bioinformatics liner, once the tool is wrapped and generated and provided into the repository, this documentation is immediately generated. And it gives you quick start guides on how you might want to use it in Janis. Because they're generally available installed automatically, you can import them into your workflows. You can directly run them from the command line. And because all of the tools have containers, you don't need to install any software to use it, except for Docker. So looking at our new execution overview with Janis, we now have one Janis spec that can generate CWL, can generate WDL, and could potentially generate some future specification, which gives us a more overall picture, more overall picture of the amount of execution engines we can use and more compute environments that we have available to us. But the problem of output and logs and errors in individual formats is still a problem. So we talked about the definition, which is specifying your workflow and specifying your analysis. The specifications we've considered are CWL and WDL, which help us describe the steps in our workflow. And importantly, the definition is abstract from the compute environment, but has inputs and outputs in a container. But now we consider how we run our workflows. We use an engine, which accepts the CWL or WDL specification, and some common examples of engines are Cromwell, CWL tool, and Toil. The execution runs on a compute environment, so maybe our local computer, our HPC environment or cloud, and it uses some container environment like Docker, Singularity, or UDocker. The only comment I'll make on the container environments are that Janis specifies OCI compliant containers, which all of these these container environments accept. So the one container that you specify in Janis can be used across Docker, Singularity, and UDocker, and so on. Now, our execution engines can be difficult to configure. So how can we make this easier? Well, we now have a Janis assistant. The Janis assistant's primary goal is to help you run workflows. It has progress monitoring, like this image on the right here. You can see that when you're running, say, a germline workflow, it can tell you all of the steps in your workflow, how long they've taken to run, and which steps are still to run. The Janis assistant can standardize the output collection process. It can also notify you when your workflow moves in between stages, such as running or failed or finished. The assistant has configuration templates for Slurm, PBS Talk, and can currently run your workflow using CWL tool and Cromwell, which are fully managed by Janis and can be changed just by simply specifying the engine parameter. And we'll use the Janis assistant in the demonstration as well. Simplifying our execution diagram, we now have our Janis specification. Janis specification, we have our CWL, WDL, or future specification, and our execution engine. But this is all managed by the Janis assistant. In the end, we have some standardized set of outputs. So whether you're getting outputs from Cromwell or CWL tool, they'll all appear the same to you. So now it's time to do the demo. So we're following the tutorials, the tutorial 0 and tutorial 1 from the Janis documentation. And these links are available at the end as well. So first off, there's some information about Janis and what we've talked about in the presentation. The requirements for this tutorial are a local environment. So we're not talking about HPCs at the moment. Python 3.6, which is a requirement of Janis, and Docker. If you don't have virtual env installed for Python, it's a pretty easy pip install. So we can create and activate the virtual env just with the following 2 lines. And then we're going to install Janis through pip. So now we've activated the virtual environment, we can run pip install Janis pipelines. And this will go and get all of the individual components of Janis. We can see here there are core, assistant, which is helping us get to run. But we also have our toolboxes, Unix and Bioinformatics, and a set of templates which help us run on HPC environments as well. So now this is just about finished. We can run this Janis V to make sure that we've installed Janis correctly. Perfect. So CWL tool is automatically installed with Janis now. And we can run an example workflow with Janis. So we talked about the tools that are automatically available to you. And one example is the hello world, which you can run through Janis, run hello. And we're just going to do that right now. And we're going to make sure that this runs with CWL tool. Great, so it's moved our task outputs into this folder here. So we can, I think I'm missing some of my tab, there we go. So I can look in our output directory and see that there's a file called out. And if I cat the output of this file, I have hello world. So now let's start building now that we've installed Janis and we've made sure it worked by running the hello world example, let's start by building a workflow. This tutorial has a set of reference data available for you. So you can just run the wget or the curl command, and it gives you the reference file, the set of compressed FASTQs, which we're going to use to, which we're going to align. So let's talk about this workflow. We're going to start with a pair of compressed FASTQ files. We're going to align these reads using BWA mem into an uncompressed SAM format. We'll compress this into a BAM file using SAMTools view, and we'll sort the reads using GATK4 sort SAM. And all these tools have attached documentation here. So we could see for BWA mem, there's the documentation available from BWA. There's a quick start guide, how you can use it in your workflow, which we're going to follow. How you might want to run this tool just via the command line. and also some extra metadata like the container it's going to use, the versions available in Janis, and also the number of ways you can configure BWA or your tool. So the inputs we're going to focus on today are reference, reads, and there's a read group header line. So we've already created the file for you. In this tutorial, we've already created the file and we've imported the different tools. So let's jump into our alignment file and have a look. Okay, we haven't imported. So we're going to import our data types and our tools from the Janis Bioinformatics repository. And we're also going to import our workflow builder and string types from the Janis core. Now workflows have, and now let's declare our workflow. So we're going to use a workflow builder and create a workflow called alignment workflow. I'm doing this in Vim, but you could very easily write this in VS Code and get more direct Python help, Python IDE support. So now that we've imported our tools, BWMM, SAMTools view, SortSam, we've got our workflow, which we've declared, and we have a set of bioinformatics data types. Let's start by creating some inputs to our workflow. Now, inputs have the format identifier, data type, and then we could also optionally provide a default or doc attribute. For this workflow, we're going to create inputs for sample name, a read group header line, which BWMM needs to help align the files, a FastQ, which will be a FastQ GZ paired end, and this just means it's two FastQ compressed files, and our reference. Now, faster with dict here is our normal faster file, but it also contains a number of extra reference things, and we could potentially look at that right now. So inside our directory, we have data and reference, which are our two folders that we downloaded from the workshop. So inside data, we can see the two FastQ files, and inside reference, we can see a cut down BWA, a cut down HG38 reference set. I can also probably make this a little bit clearer. So beyond just the normal faster, we have a .dict, .amb, .ann, index files, and so on. And this faster with dict reference type will make sure that Janis collects all of these references and provides them to the tool. So going back into our tool, let's declare our 4 inputs. So now that we have our workflow, we have our inputs, let's start building some steps. Steps have a very similar structure to our inputs. They have an identifier, they have the tool, and there's some other optional information like how we might want to scatter or basically loop over a tool a number of times. When we specify our tool, that's where we'll specify all our connections and dependencies and what the tool needs to run. So our first tool we're going to use is BWMM, which from the documentation we saw before, that our reads are going to expect a FastQ GZ pair, which is equivalent to our paired end. It's going to expect a read group header line, which is a string, and that's going to be equivalent to our read group, and a reference, which is our faster with dict, which we're going to connect to our reference input. And we can see this in the following step. We have a step called BWMM. It's going to use the BWMM latest tool and provide 3 connections. So we can copy that step in now. Our next step we're going to use is SAMTools view. So BWMM has, and we can see from the documentation, is going to output a SAM file. And now we need to convert it to a BAM. So we'll use SAMTools view, use the SAM connection, and use the output of BWMM. So we can jump that step into our workflow. And now we have the output of SAMTools view is going to be a BAM file. But now we want to sort it, and we also want an index file. So what we're going to do is we're going to use a GATK4 SortSam. Our input of the tool called BAM is going to be the output of SAMTools view. But SortSam has some additional options that we want to specify, like the sort order or whether we want to create an index. And we can just use literal Python values here. So we can say the sort order is coordinate, and our crate index is just going to be the value true. So we can copy this step definition here now. And now our workflow has 4 inputs, 3 steps of which are chained together. And we can see BWMM, SAMTools view is using the output of BWMM, and GATK4 is using SAMTools sortsam is going to be using the output of SAMTools view. Now we're almost there. All we need to do is output the result we get from sortsam. Outputs have a very similar structure as well. They have an identifier. We can optionally provide a data type, but if we don't provide a data type, Janis will infer this from the output we're specifying. So we're going to use the source of sortsam. And from here, just like with this, we're going to call our output out. And because we specified the source sortsam, Janis will automatically know that the output of sortsam is an index BAM and annotate our output with this type. So now we've specified our total workflow. We have our input, steps, and output. And hopefully it should look like the following Python script. So now that we have our script, and it's in a folder called tools, so we can just confirm that, we can translate this workflow to WDL with Janis translate our workflow called, which is in alignment, and we can convert it to WDL. Ooh, looks like I have a little error. Unexpected keyword argument. All right. Looks like I have, not sure here. Let me just see if this works. Okay, so it might be a little problem with my installation that I pulled from pip, but never mind that for now. So we've translated our workflow to WDL. So we've imported, we can see our import statements. We can see our WDL workflow with a set of inputs. Importantly, we can see that the reference type that we specified, which we know has a set of index files, have these separate reference inputs for WDL. So that, and then when we run our workflow, Janis will automatically fill these references in. We can see that for the call for BWMM, it's also connected all of its associated references together. And we can see that our output, as we expected an index BAM, it has the out and the index for the BAM output. We can also see that our literal values for sort order, coordinate, have been provided to sort order, and an input has been provided as well. And an input node has been created and stepped to it. We can do the same for CWL. So we can just simply convert our workflow to CWL. Now, CWL is a little harder to read, but we can see our inputs, FastQ, regroup, reference, and we can see our associated secondary files. We can see those sortsam literal values. We can see our steps, and we can see how verbose CWL can be. Cool, and now that we've translated our workflow to CWL and WDL and made sure it works, we can run the alignment workflow. So here we're going to Janis run with, we're going to use the current directory as an output directory. We're going to run it with the CWL tool engine. We're going to run the workflow that we just created, and we're going to provide our inputs directly on the command line. So we can copy this block in. If I can work out how to copy. Cool. So Janis run, and we'll see immediately some output from CWL. So it's exporting our workflow and now called CWL tool to run. And we can see BWMM, SAMTools view, and now it's running sortsam. And now the workflow has finished running with CWL tool. So we can see the outputs, and we specified the current directory as the output. And we can see our data and reference folders from before. But now we have a new out.bam and out.bam.bai, which are the outputs of our alignment workflow. Now we can run the same workflow with Cromwell, all by just changing the engine from CWL tool to Cromwell. So we can take the previous run command. We'll change the output directory to be called run in Cromwell. And we'll specify the engine Cromwell, and we'll let that run. Now, one thing we can do as well is if we jump into Ravix Composer, we can see our Janis tutorials folder here. If we look in the Janis folder, which was created by Janis to run the CWL tool workflow, we can jump into workflow and we see our CWL file, and we can double click that and see our workflow that we created immediately visualized. And this is one of the powers of using Janis, that the workflow that you create, you can use the community tools like Ravix Composer to generate this visualization. Now, Cromwell can be a little bit slow to run sometimes. So we'll leave that there for a little minute. Actually, what we can do is we can see here that Janis has given this workflow an ID called EEAF6B. Now, what we can do is while this is running, I'm just going to open up a new tab for Janis, a new tab in Terminal. And I'm going to run Janis watch and this workflow ID. So when I run this workflow ID, I can immediately see that we're using the Cromwell execution engine. I can see the task directory that we specified as an output. And I could see the progress of my workflow run. So I can see BWA mem ran for 9 seconds, SAMTools view ran for 7, and SortSam ran for a little bit longer. And then we have these sets of outputs here. So I can just ls the directory that we specified as an output. And I can see the out.bam and the out.bam.bai. So even though we ran our workflows in 2 different engines, CWL tool and Cromwell, I get exactly the same format for the output. Perfect. Let's jump back in here. Cool. So in summary, we installed Janis in a virtual environment. We ran the Hello World, which is a tool available in the toolbox. We ran that directly from the command line, and we didn't have to install any software. We built an alignment workflow using Janis, in which we imported tools from the registry, like BWA mem, SAMTools, and SortSam. We declared a set of inputs, steps, and outputs on our workflow, and we translated and ran this workflow in CWL and WDL. But we also ran it with Cromwell and CWL tool and got exactly the same results. We got exactly the same outputs. So some examples of what you can do in Janis. We didn't do this in the demo, but you can initialize your environment to use a Slurm singularity, or there are a number of pre-built templates that are available for Pawsey, PeterMac, Melbourne, Spartan, and WEHI. We saw how you can translate an existing workflow or tool to WDL. We can run a tool with directly from the command line with Janis run, and we can monitor the progress, which we saw with Janis watch and that workflow ID. So the tool types that are available in Janis are the command tool, which is how we've interacted with command line programs. A workflow is also a different type of tool, which is a combination of these tools. Workflows are mainly used to encapsulate logic into modular components. And because workflows are a type of tool, you can embed workflows into each other. One other tool type that's unique to Janis is called a code tool. So we currently have the Python, which allows you to portably run a Python code snippet in your workflow. So rather than writing a block of Python code, putting in a container, working out how to build and publish the container, and then re-annotate it in Janis or your CWL or Cromwell, you can just fill out a class, a block of Python code with your script, and it will immediately run in all the containers portably. So one thing we'd like to highlight as one of the ways that Janis can abstract some of the complexities away from these languages is scattering. For example, if we had the workflow with the inputs myBAMs, which would be an array of indexed BAMs, and an input called references with an array of indexed references, and then we had some step that took those two, but that only expected a single instance. So my tool is expecting just a single BAM BAI and just a single faster BWA. We could scatter over those two inputs using the dot product. And in Janis, it's as simple as just by scattering with this array. But in WDL, you would have to, this would translate to the following workflow. So you would have an array input of my BAMs, and also for its indexes, an array for the references and the references indexes. But then you have this pretty nasty scatter statement and the mappings that go into my tool. And this is just one of the ways that Janis can abstract some of that complexity and allows us to make specifying workflows simpler. So we talked about Janis being extensible, and maybe you have tools that you're not ready to publish yet, but you want to use them in Janis. So, well, Janis has support for private toolboxes. So all you need to do is specify a set of entry points in the Python module, and you can create your own execution templates and data types and tools just with these extra entry points. So if I have my Janis workflow, how would I potentially move my analysis to the cloud? So say you had this example where I'm running with some local configuration with some local set of inputs and my workflow. The only thing you need to do is change to a cloud configuration, and in your inputs YAML, add the GS prefix and basically reference the cloud paths to your inputs. Your pipeline can stay exactly the same, though you might want to make changes to be cost efficient. With Janis, we've produced a number of exemplar pipelines, mainly for whole genome sequencing for germline and somatic variant callers using the GATK4, Strelka, Vardict, and GRIDS variant callers. And we can see a visualization of this on the right using the three variant callers that we get from Ravix Composer. These exemplar pipelines demonstrate that Janis is a functional framework that can produce valid and workable workflows. And these exemplar pipelines are available publicly. So you can see them on the documentation, you can run them, and you can run them directly from the command line as well. We've tested these exemplar pipelines on a number of compute environments, like a personal computer, the Peter Mac cluster, UniMelb, WEHI, Pawsey, and also Google Cloud across a variety of workflow engines, a number of batch systems, so Slurm, PBS Talk, and the Pipelines API for Google Cloud, and a number of container environments as well. And we've ensured that we get perfect reproducibility across these environments. We're using Janis and Peter Mac for these cancer variant calling pipelines. And because we've produced them in Janis and we get those artifacts, the CWL and WDL, which are publishable artifacts, we can share these pipelines with other researchers, such as Spun. We can make our in-house pipelines more portable, which means especially during the COVID times, we can run our pipelines on Nexta instances, and we're also performing a trial analysis on GCP. There's a researcher at Peter Mac who's running multiple tumors and normal on the Cascade project, who's running multiple 160x tumors paired against a single 100x normal, and doing some pretty heavy scattering. So after 1,200 genomic regions, so generating a lot of jobs. And interestingly, these intervals are dynamically generated in the workflow, within the workflow. And he's running the bleeding edge of tools, so very constantly iterating on the tools that he's using. WEHI are doing a tumor normal analysis, querying the genomics data commons with this exome data, and then running a Janis pipeline for each tumor normal pair with a number of copy number callers. there's about 5,000 exome tumor normal pairs analysed, and they take anywhere from a portion to up to 3 days to analyse. Some of the future work for Janis we're looking at is expanding the toolbox, building new pipelines outside of the cancer domain, and supporting new users. And we're doing this by improving documentation, by expanding the toolbox, but also being available on GitHub and GitHub for questions. We're also wanting to make it easier to configure Janis for new compute environments. So in summary, portable pipelines are extremely useful. We've produced Janis, which is a Python module for building workflows. It can generate CWL and WDL. Along with Janis, we've produced an assistant to make running pipelines and workflows simpler across a number of compute environments. And today we also demoed how the process of building an alignment workflow and running and how building this with Janis allows you to immediately get CWL, WDL, and also run it on Cromwell or CWL tool. I'd like to thank members of the Portable Pipelines Project, Richard, Evan, Danny, Benny, and John for being there for constant feedback and questions and allowing us to quickly iterate on the different ideas. The steering committee for making the project possible, especially seeing as it's a collaboration between some pretty large institutes. The initial proof of concept for generating CWL and WDL and being more flexible, and a number of other contributors who have helped build tools and run and test workflows. So how do I get going? Well, we demonstrated how you can install Janis, but it's immediately available from pip. There's a number of tutorials and workshops that are available online, and really Janis is as simple as importing Janis into Python. I'd like to direct you towards the documentation, the GitHub, where if you're having issues or questions, you can also jump on Git as well. And thanks very much for listening.
Thanks very much for a great presentation, Michael. It's now over to you, the audience, to submit any questions you'd like Michael to address before we wrap up today. Now, Michael, you have nicely demonstrated how Janis can simplify the building and running of workflows, and also demonstrated that you've got some really significant partners finding benefit from using it. I'll mention that Australian Biocommons found Janis so promising that from July, we will co-fund its further development as part of a command line interface activity within our ARDC platforms project. And that's really with a view to finding users nationally who can benefit. Now, we hope some of those people are listening in today, but you've been doing presentations all around Australia, Michael. Have you had good interest in the project?
Yes, so we presented at Abacus in December last year, and we found that a lot of people are starting to run into the problems that we're facing with sharing the pipelines and making them available on different platforms. So it was great that we've gone through some of these processes of containerizing tools and looking at the advantages and disadvantages of making Cromwell work with Singularity on Slurm or PBS, and we've just helped to pave the way for people doing that. So I can see a couple of questions. So I'll answer the first one. So once you've written a workflow for Slurm Singularity, does Janis provide a way to translate it to other schedulers or container engines, example PBS? So there's two sides to this question. First, the specification. So writing the workflow doesn't interact with Slurm at all. So you just specify the jobs that you want to run, and then the execution engine like Cromwell or Toil or Nextflow, if you're using Nextflow, can pick up those jobs, and the actual engine knows how to submit it to PBS or Slurm. So to answer the question, the Janis core workflows that you build will run with PBS and all of the schedulers that your engine can provide. And we're looking at producing more templates for the assistant, so that it's very easy for the assistant to configure Cromwell to submit for for new schedulers. And there's another question here. How does Janis handle feeding in multiple input data at the same time? Oh, what's this prompt? Let's come up. Okay, I see. So you can specify multiple inputs to Janis using wildcards on the command line, as we did actually in the example. So the example collected all of those. I wonder if I can close this. So the actual example did use a wildcard to collect all of the FASTQs. But if you have multiple sets of samples, maybe I want to align multiple sets of FASTQs together. Janis actually has a batch run functionality, which I can put a link to in the webinar chat, which allows you to basically, run lots of different, run the same step multiple different times, all from the command line without having to do any kind of workflow wrapping it around it. And the outputs are organized. There's a couple ways to do this, but there is a tutorial online. But actually in the output section, you can see there's an output name and an output folder that you could specify for an output. So if you have a bigger pipeline, like say our germline pipeline, you could organize all the BAMs into a folder. You could give all of the specific outputs a name, or you can reference maybe the sample name that you want to name your output file. We're trying to make it super flexible that if you have to interact with another system after Janis, that you could prepare your outputs in a way that just makes it really seamless.
We've had another question come in there too in the chat box. It's related to error handling and snapshots for resume after errors.
Yeah, okay. So the Janis assistant is a layer on top of Cromwell. So we've added some extra things into that to help with the error handling process. So essentially, if your tool fails, and then Cromwell will catch that and report the error back. I don't think I have the slide where it says error anymore, but jump back to the progress screen. So on this tool here, on this screen here, it would say there'd be a red X that said your tool is errored here. The workflow would move into a failed state, and you would get a standard error on basically what happened to the tool. And then all you would need to do was maybe tweak your inputs to fix the tool. You would Janis run on the same folder on the output, and then you could use the previous results to automatically jump to halfway through. So I'll make that more concrete. So for example, if mark duplicates here failed, and I had to make some tweak to fix mark duplicates, when I reran my workflow, it would say, oh, well, I've already actually computed the results for cutadat, BWMM, sortsem. So it would skip all of those and it would jump straight to mark duplicates. I hope that has answered the resuming question.
Great. Well, thank you so much. I think that's all we have time for today. So once again, I'd like to express our thanks to you, Michael, and I'll draw the presentation to a close.
Thank you. Thanks for having me.
We'd like to acknowledge that our funding at the Australian Biocommons, we're enabled by NCRIST, primarily via Bioplatforms Australia funding. Thank you so much for watching today. We hope you found it useful. And until next time, goodbye.